concept of an antibody can seem really abstract, but really they're just little proteins, and from a biochemist perspective, great tools in the lab. For doing things like a Western blot, where we're looking to detect specific proteins on a membrane after we run an SDS page gel. So basically, we separate proteins by size in a gel, we send them onto a membrane, and then we use antibodies to probe the membrane to see if specific proteins are present. We can do something called an ELISA, which is similar in concept, except that things are happening in the well of a plate rather than on a membrane. We can do things like a co-immunoprecipitation, a co-IP, a pull down. Basically, we use an antibody to pull down a molecule of interest, a protein of interest, and then see what it's bound to. All sorts of very cool things that we can do with antibodies. So today, let's talk about the structure of antibodies, a bit about their biochemistry, and how we can take advantage of that biochemistry to learn about other biochemistry. We'll talk about the theory as well as see some examples of how to interpret the figures that they produce. So let's go. I like to think of antibodies as kind of a biochemical version of Where's Waldo, where basically you have these little proteins that are able to go search for specific unique qualities of a specific protein, or it doesn't have to be a protein, but some sort of thing that this antibody recognizes. And when we say recognize, really all that it means is that that antibody is going to bind, hopefully very specifically, to something. And we'll talk a little bit more about how this actually occurs. But basically, there are parts of the antibody that is kind of like generic for the animal that makes it. And then there's also these like variable regions, and these variable regions are going to be the parts that actually bind to the thing that it quote unquote recognizes. So that thing that it recognizes, we call that the antigen. So Waldo would be our antigen, and the specific part that it recognizes on Waldo. So the specific part it binds to, that is called your epitope. So we have the antigen, which is the thing that the antibody recognizes, and the epitope, which is the exact part of the thing that the antibody recognizes. So the part that it's actually binding to. And these each antibody, each like different antibody is going to have a different antigen binding site. And these are going to be on the most typical type of antibody. We're going to talk about this IgG antibody. They're going to be like on the tips of the Ys. And those are going to be the parts that are going to bind to that epitope. And we'll talk much more about the details of the antibody structure, as well as how it actually goes and does that recognizing, does that binding, because we'll see that it's just going to involve those non-covalent interactions, all those good IMFs that we've been talking about. Yes, it's even going to include that hydrophobic effect. See, I told you it's all connected. I told you biochemistry was all around us. And by better understanding biochemistry and how antibodies work, we can do things like design new antibodies, produce antibodies, and take advantage of antibodies in the lab to study all sorts of other things. And so that's what we're going to be talking about today, is how we can use antibodies as tools. For example, we can go and we can see if a protein was being expressed. So if cells were making a protein. We can also isolate that. If we use antibodies against a protein, we can kind of use that to pull out that protein. So if we attach those antibodies to, say, beads, we can do this thing called a co-immunoprecipitation, where basically we use the beads, we like pull on the beads, such as with a magnet, and then this is going to capture our protein as well as anything else the protein is bound to. So we can go and we can look and see what that protein was binding to. So we'll look at examples of how to like interpret the results of these, as well as the theory behind how they work. So we'll talk much more about these methods, but basically what you're looking at the left is something called an ELISA, and we'll get more into this, but it's an enzyme-linked immunosorbent assay. Basically, you have an antibody or you have an antigen, which remember is the thing that the antibody binds to, and one of them is going to be attached to the surface of a plate. And then you can add the other thing on top in some sort of like mixture. This can be a blood sample, this can be some sort of solution, and then see if they bind. So we'll talk more about this, but that's an ELISA. On the right, what you're looking at is this is a Western blot, and it's AKA an immunoblot. So you might see that term used. And we'll talk about how we can use antibodies to recognize specific proteins after we run an SDS-page gel, like those gels that we talked about in the lab and which you guys are going to do. We're not going to do a Western blot, but it is good that you know how they work, especially because that's going to help you understand how co-IPs work and how to interpret their results. And so again, we're going to be going over a couple examples in this video, and then we'll be going over a lot more in class, and you guys will get practice working with them and interpreting the figures. But let's start with what is an antibody. And so when you hear about antibodies in the news and all of this various things, it's kind of like this abstract topic, and you're like, okay, it's a little thing. I don't really know what it does, but it helps protect me from disease. Well, antibodies are really just little proteins. And basically, they're made up of these 4 chains. So they have these 2 heavy chains and these 2 light chains. You don't need to worry about knowing all the specifics about the antibodies, but know that basically they are going to have these chains that are connected through disulfide cross-links. So those disulfide bonds. So remember, when we talk about disulfide bonds, what are we talking about? Right, we're talking about those cysteine-cysteine cross-links. So remember, cysteine, our good old friend from our amino acids. So remember, you can't forget these guys. We got to keep remembering them because they're going to show up over and over, because these amino acids are basically like our biochemists or our cells toolbox. These functional groups from the amino acids are going to provide the tools for doing things like binding. And when we talk about enzymes, they're going to be doing things like catalyzing reactions, speeding up reactions, playing key roles in making life possible. So amino acids, super important to know. And so what do we know about cysteine? Well, cysteine is going to have that thiol as its side chain, in its side chain, that SH group, and it can oxidize and form cross links. So if you have 2 cysteines and you have an oxidizing environment, then what can happen is that they can form these cross links. And we talked about how the environment inside of cells is really reducing, but the environment outside of cells is very oxidizing. So why might antibodies have a lot of disulfide bonds? Yeah, these antibodies are going to be secreted, or they're going to be on the outside of cells. They're going to be in that oxidizing environment where those not only will those crosslinks survive, but they will also be able to kind of certify those antibodies and allow them to travel through the blood and all of this stuff without just falling apart. But if we're in the lab, how would these antibodies be affected by the presence of DTT, or beta-mercaptoethanol? So think back to what these are. Remember, in the lab, we used DTT when we were purifying our protein because we wanted to mimic the intracellular environment. So how would adding DTT to an antibody affect things? Yeah, the DTT, it serves as a reducing agent. It would break up those disulfide bonds. And so if you were to, say, run an antibody on an SDS-page gel that you added a reducing agent to, like BME, then you would see those chains would run as separate proteins. But if you didn't have a reducing agent, you would see a single bigger band. And so we'll talk more about this. So remember, the IgG is the kind of most common one that we're going to be seeing, and it's got this Y shape. What we can also talk about is you might see something like the FAB, the fragment antigen binding, or the FC, the fragment crystallizable. Basically, they're just parts of the enzyme. So the FAB is kind of like one of the arms of the Y, and then your FC is kind of like the bottom part, because basically there's this part that if you cut it off, cut it there, then there's this fragment that was easy to crystallize, and so they called that the fragment crystallizable, or the FC portion. But the FAB portion is actually going to be the part that binds to the antigen. So don't worry about remembering FAB or FC, but do know the terms antigen, which is the thing that the antibody binds to, epitope, which is the exact part that the antibody binds to. And know that antibodies are going to have these kind of like variable regions and these constant regions. So there's a variable and a constant region for both the heavy and the light chains. So what else can we tell by looking at the structure of an immunoglobulin and these IgG antibodies? Let's think back to the levels of protein structure. See, I'm not going to let you guys forget the stuff that we learned before, because the reason why we learned it is because it's all connected, and it's going to help us understand
all of these later concepts. So let's think back to our levels of structure and think about how many levels of structure we see in an IgG antibody. Yeah, we see 4. Basically, we have the primary structure, which is just a sequence of amino acids. We have the secondary structure, which involves backbone hydrogen bonding and gives us things like helices and strands and sheets. We have the tertiary structure, which involves side chains and the single chain, the structure of a single chain. And then we have quaternary structure because we have multiple chains. So we have these 4 chains. With each of these chains, we can basically have a tertiary structure. And then together we have the quaternary structure. What is the main type of secondary structure that we see in IgG antibody? Right, we see beta strands and beta pleated sheets. We don't really see any alpha helices, but we do see a lot of those beta strands and beta sheets. And in fact, antibodies have there's this like IG fold, which is kind of this fold that you find one of those motifs that you're going to find in these different in these different chains. So now that we know a little bit more about the structure of antibodies, let's talk about how they're actually made and how they actually bind. So antibodies are made by a type of immune cell called a B cell. So I am not an immunologist and I do not know all the complicated details of immunology, but this is just kind of like a basic overview. What happens is that you have these B cells, and these immature B cells have a whole menu of options to choose from when deciding what antibodies to make. So basically, the genes for making antibodies, they have a bunch of options. And then the cells, they kind of edit that DNA. So each cell kind of makes a different random choice about how they edit and remove parts of that DNA and piece other parts together in order to make a unique antibody. So each of these B cells is going to make a unique antibody, just like randomly choosing. So they make different initial choices, and then this determines what it makes, and what it makes determines what it can bind and whether it can recognize an antigen that that the animal or the organism is confronted with. So remember, these are just randomly chosen. So they basically mix and match pieces, and there's going to be tons and tons and tons of different versions of antibodies. And so some of them might not recognize anything that you're ever going to encounter in your life, but some of them might. And so if you encounter something and one of those antibodies successfully binds to it, so it sticks to that thing and also doesn't stick to your own body, part, so it doesn't recognize yourself as foreign, which would be bad. So your cell gets rid of that. But if it recognizes that foreign thing, then what's going to happen is your body's going to mount an immune response, select for that cell that made that, say, oh, this one made a good one. Let's make more copies of it. And so that cell that gets selected for, basically you make lots and lots of copies of it. Some of those copies get secreted. So some of the B cells that come from that initial cell, we call these effector or plasma B cells, they start secreting that antibody. And other cells, memory B cells, kind of keep it sticking out for the surface of the cells. And together these are going to do things like help protect you from the invading virus, remember it in the future, and also they can go and undergo this thing called somatic hypermutation, where basically they made that initial choice about how to mix and match the pieces, but then they can kind of edit it further in order to gain better affinity. And then we can take advantage of these in the lab. But typically, when we want to use the antibodies in the lab, we're not wanting to use antibodies that are, say, against a viral protein. I mean, we might be in some cases, but often we just want to study all sorts of different proteins, and sometimes not even proteins, sometimes modifications on proteins, sometimes random other things. And so we need a way to do this, and antibodies can actually be made in a variety of strategies in the lab. The kind of classic strategy is that you inject an animal with something that you want to act as an antigen. So remember, our antigen is the thing that the antibody binds. And then that lab animal is going to make antibodies. Now, just like when you make antibodies in your body, some of them are going to be pretty bad and some of them are going to be pretty good. And so basically, your job is you're going to want to isolate those really good ones if you can, Or if you can't, you can kind of just isolate all of them that bind. So basically, the antibodies are going to get secreted in the bloodstream. And if you just like collect all of those antibodies that bind to a specific antigen, what you get is this thing called a polyclonal antibody. It's called a polyclonal because it's coming from, like, we call it like a clone, like a B cell clone, basically one cell that makes lots and lots of itself. If you have lots of cells that are making different antibodies that all recognize that same epitope, that all recognize the same antigen, but at different epitopes, so different places, then you can collect those and you get what we call a polyclonal antibody mix. If you want just those really good ones, what you can do is you can isolate the cells that make those really, really good ones. And then you can either do a thing called like a hybridoma, where you basically get the cell to make lots of copy, like to grow forever and make lots of copies of it. Or you can actually nowadays take that genetic information for making that antibody, stick it into cells and get cells to make more with all those good recombinant protein expression techniques we've been talking about. And nowadays, there are even techniques that don't even involve lab animals in the first place, to kind of try to evolve antibodies in the lab directly through like expression systems. So lots of different ways, and then we can take advantage of them. But even with having those new strategies and things, there are lots and lots of different proteins that people want to work with. And these strategies take a long time, they're expensive. And so instead of needing an antibody for every single protein that we want to study, what if we can just have an antibody? What if we can use an antibody that recognizes a tag that we put on our protein? So remember how we could use tags to affinity purify our protein? We could stick on something like a GST, we could stick on a his tag, we could stick on a strep tag. And those would allow us to purify out the protein by using beads that are attached to something that binds to that tag. Similarly, we can use tags on proteins as epitopes for binding antibodies. Then what we can do is basically we just need to use an antibody that recognizes that tag. We don't have to use an antibody that recognizes a specific protein. So in this case, the work that we're doing, instead of paying to like get all fancy antibodies, what we're doing is instead we're just going to be somehow adding a tag to the protein using some sort of recombinant method. It's harder when we're doing things in like your eukaryotic cells, but there are ways with like viral vectors and things like this, CRISPR, all sorts of different strategies. You can introduce a tag. When you introduce a tag, well, now you have a version of the protein that you can detect. Not only can you detect it, but you can also pull it out, see what it's bound to, and things like this. Now, for when you're using a tag as an epitope, you typically want to use something small, so it's not going to interfere with the protein. So some of the tags that you might see are like HA, flag, MIC, V5. Sometimes people use a his tag, these short little tags that basically, they shouldn't interfere with the protein function, so you don't have to worry about like cutting it off. But they do allow for antibodies to detect them. Sometimes these tags, I don't know why it auto-corrected to Tages, that's weird. But basically sometimes these are also known as haptens. So they're just these little tags or these little things that you put on your protein or whatever else you want to be able to be detected by an antibody. And again, when we're doing this, we can basically use antibodies that are polyclonal. So they would be recognizing different sites on Waldo, so different epitopes, or monoclonal, where the antibodies, all the antibody copies in there are all recognizing one epitope, so one site. So in the lab, we're typically using these IgG or these immunoglobulin antibodies. But in our bodies, we also make these things called IgM antibodies. These are actually made earlier in the infection. They have a weaker affinity, but they kind of like team up together and they form these pentamers. So there's 5 antibodies. And so they're connected through disulfide bonds and with this thing called a J chain. And what happens is that having all of those together allows for this thing we call avidity. So basically what happens is if you have multiple binding sites on something, if one site binds, then it's kind of easier for the things nearby it to bind because the first thing's kind of tethered things in place. So remember that if you have things floating all around, it's going to be hard it's going to be really hard for them to find each other in the first place. But once they find each other, then you have these sites. If you have sites nearby, you can get additional binding. And this is going to allow those weaker antibodies to compensate because they're working in teams. But then what happens is over time, you start developing those IgG antibodies, and especially due to somatic hypermutation and stuff, where they're kind of editing to further refine how good those antibodies are, you can end up with antibodies that have very high affinities. So we're talking KDs on the order of 10 to the minus 12. So when we talk about 10 to the minus 12, well, that's going to be like a picomolar. So remember, 10 to the minus 9th is going to be our nanomolar. And we're going to be talking about somewhere, like sometimes we can have ones that are on the range of picomolars. So these antibodies can have very, very high affinity. So remember what the KD is? The KD is telling us the concentration of one binding partner at which half of the other binding partner is bound. So in this case, it could be talking about the concentration of the antigen at which half of the antibody is bound to the antigen. So remember that the weaker binder is going to have a higher KD. So the KD is the concentration at which half of it is bound. And so you can remember that things are just kind of like floating around randomly. And if they have a high and bumping into one another. Now, imagine that they bump into one another and they like each other. Well, then they're going to stick. And so even if there's not that much, even if the bumpings into each other, the chance encounters, even if those are fewer, you're still going to find a lot of it bound because they're sticking tight. But if the binding is weak, you're going to need more of those chance encounters so that even if they don't bind strongly, even if they bind and let go right away, there's going to be another antibody or there's going to be another antigen, there's going to be another ligand right next door to kind of then go bind, even if transiently. So you'll need a higher concentration, but eventually you will reach that same half saturation point, half proportion bound point. So here's an example from a paper where they're basically looking at the ability of antibodies to bind to SARS-CoV-2. And what they can see is, so basically they have cells that have these that are displaying this viral protein, and then they have these antibodies. And they can test different amounts of the antibodies and then compare how much of the cells are bound to them. So what we can see here is, like, although this isn't in molarity, we have this basically on our x-axis. This is going to be a concentration of our antibodies. And on the y-axis, we're talking about the proportion bound. Well, remember that what is going to have the stronger KD? Is it going to be to the right or to the left? Right, a stronger KD is going to be to the left because this is going to indicate that less of it you need less of it in order to get half of the binding. And so what we can do is, if we go over here, we can see, okay, this is going to be our strongest binder, our S306, whatever that is, and our weakest binder, that's going to be this S124. And so that's kind of like how you would interpret one of these curves, is by looking to see, okay, what's most to the left, what's most to the right. We can compare between them. So we can say that this one is higher affinity than this one, and things like this. We can also basically just remember that this is on a log scale. So we can't tell anything about, like, by the shape of this curve. This is not telling us it's cooperative binding, because everything is going to look sigmoidal on a log scale. And when we're dealing with something like this, this is basically just showing us the binding curve, nothing about cooperativity. And remember that the KD is also going to come from our rate constant. So it comes from the rate constants of binding, which we call our K on, and the rate constants of unbinding, our K off. So remember, KD equals K off over K on. The KD itself does not tell us about the kinetics, so whether it's has a higher off rate or a lower on rate, or a higher on rate or a lower off rate. We can't tell that from the KD alone, from equilibrium measurements, but we can tell it if we actually go and measure binding kinetics. And so when we on class on Friday, we talked about different ways we can measure binding affinity, SPR, BLI, etc., and how some of these techniques like SPR and BLI actually allow us to measure binding kinetics. So remember BLI, biolayer interferometry. This is where we kind of have this sensor, and on the surface of the sensor, we have one of our binding partners attached. So we might have our antibody attached to the sensor. And then what we could do is we could then dip it into a solution containing an antigen, so containing whatever antigen we want to look for. And then what can happen is that when the antigen binds to the antibody, it's going to change the way that light interacts with the surface of the sensor, and we can detect this change. So from something like this, we can get basically the KD, but we can't tell anything about whether the K on or the K off. But we looked at this other example in class where basically they were using BLI to measure the binding rate constants, so our K on and our K off, with a variety of different monoclonal antibodies. So remember the monoclonal antibodies, you actually isolate individual lots of copies of the same antibody, recognizes the same epitope. Then they can do that BLI measurement in order to figure out the kinetics. So basically they can put the sensor into the solution to measure the binding, and then wash it off to measure the dissociation. You can see that some of these have a really fast dissociation. This one, super duper fast, K off. And some of them, however, have a slower dissociation. So these are going to stick around longer. And the K-ons can also vary a bit, but they tend to be more similar. But let's look and let's compare. Okay, so we said that this like BLN4 and BLN6, these look like they have really fast K-offs, whereas BLN1, it looks like it has a slower K-off. So remember that KD is going to come from K-off over K-on. So how would we expect the KDs, if their K-on is similar, how would we expect their KDs to differ? Yeah, we would expect the BLN4 and 6 to have a higher KD, a lower affinity than the BLN1. And if we go and we look at the chart where they've actually calculated the KD based on their K-on and K-off, we can see that the BLN1 has a KD of 0.7 nanomolar, whereas BLN4 has 34, and BLN6 has 15.8. So these guys are going to indeed have a higher KD, a lower affinity than our BLN1. So basically you can get the affinity data from kinetic data, but you can't get kinetic data from simple affinity data. So what determines this affinity? What's determining why some of those are really good binders and some of them are really bad, not so good binders? Well, it all comes down to our thermodynamics, remember? We have that same equation we've been looking at over and over and over. Our change in delta G, our change in free energy is going to equal our delta H, so our change in enthalpy, minus T delta S, our change in entropy. Whether the binding is thermodynamically favorable, well, what do we want in order for something to be thermodynamically favorable? Do we want delta G to be negative or positive? Right, we want it to be negative. This is telling us that basically after binding, we have a lower energy state. We're at the bottom of the hill rather than a higher energy state, the top of the hill. So a negative delta G is good. In order to get a negative delta G, well, we have to think about enthalpy and entropy. So we have to think about delta H, are our new bonds and interactions more favorable than those ones? If so, we're going to have a negative delta H, which is going to be favorable. We also have to consider our change in entropy. Do the molecules in the system have more or less freedom after the binding event? And remember, this is where it gets confusing because you would think that every time things are binding, you're going to be losing entropy. But that's only when you're thinking about just those two binding things in isolation. Yeah, they're going to be now constrained more because they're bound to one another, but you might be releasing a lot of water. So binding of antigen to antibody is non-covalent. What types of interactions might this entail? Right, so it's going to entail all of those IMS we've been talking about from the very beginning. We can have ionic bonds, our salt bridges, which are going to be the strongest, but require there to be full charges, full opposite charges. We can have hydrogen bonds, which are a special type of our permanent dipole. And we can have other transient dipole-based attractions, London dispersion forces, all of that good stuff. So all of those can contribute to our enthalpy. And we have to consider the interactions between the solvent and the binding partners, as well as between the solvent molecules themselves. When antibodies bind to the antigen, it's often through this thing called induced fit, where basically the antigen binding site isn't fully like exposed or developed until the antigen actually binds it. And then when the antigen binds it, the protein kind of shape shifts a little. So remember, when we can call these shape shifts conformational changes. And sometimes these conformational changes can be really dramatic, but in the case of an antigen binding to an antibody, typically they're smaller, but they can still have important effects. So let's look at the structure of part of an antibody bound to an HIV fragment antigen. So this is that like FAB fragment, that Fab fragment I was talking about, basically the tip of the Y, the part that actually binds to the antigen. And so here what we're showing is basically we're showing this FAB fragment colored by hydrophobicity. In yellow, we have the hydrophobic regions, and in blue, we have the hydrophilic regions, and green is somewhere in the middle. So what can we notice? Well, as we might expect, the hydrophobic regions are kind of going to be more in the center. Over here, we can see that basically there are hydrophobic regions that are kind of exposed, but not nearly as much in the center figure, which is where the shape of the antibody after the antigen is bound. But so in the center, they're basically not showing the antigen, but on the right, they are showing the antigen. So they're showing here the antigen binding pocket after the binding event and before the binding event. What can we notice is different? Yeah, it kind of opens up more and exposes more of that hydrophobic surface. So you can imagine that if you had a lot of hydrophobic surface exposed all the time, that would make water really, really mad, and water might kind of like force these things together through that hydrophobic effect. So this might just clump up and make the antibody insoluble, which would be bad. But by exposing it when the antigen binds, well, in this way, you're able to kind of like safely open it up to have that binding event. Now, what types of properties would you expect this antigen to have if the binding surface looks like this? Right, we would expect it to be hydrophobic. Because of the hydrophobic effect, where basically what's going to happen is the water molecules are going to try to make as many water-water interactions as they can. They can't make favorable interactions with those nonpolar things, but they can make lots of favorable interactions with other water molecules. So they're going to try to maximize their water-water interactions. And this has the effect of drawing together those nonpolar things, kind of clustering them, freeing the water molecules that had been surrounding them, leading to a big increase in the entropy. And then once those nonpolar things are drawn together, then you can get those dispersion forces and things like this, have favorable interactions, contribute to your enthalpy factor. So in this case, the antigen was hydrophobic and the binding site was hydrophobic. But this is not always going to be the case, because remember that each antibody is going to have a unique FAB fragment. It's going to have a unique variable region. It's going to have a unique binding site. And so sometimes you might have a basic binding site, which would want to bind something acidic. You might have an acidic binding site that might want to bind something basic. You might have a hydrophilic binding site, or you might have a hydrophobic site, like in this case. But in all these cases, you're often going to have this induced fit mechanism, where basically the full shape of the protein is going to change a little after it binds to the antigen. Not nearly as dramatic as what we saw with hemoglobin and stuff, but it can still undergo a shapeshift. And keep this concept in mind of induced fit, because it's going to come into play when we talk about enzymes and how enzymes kind of use induced fit when they bind to substrate to help get that substrate in the right shape and stabilize the kind of tipping point rather than stabilize the substrate as it is. Okay, but now let's go and let's talk a little bit about techniques. So let's start with our Western blot. So remember in lab, we talked about SDS page and how we could basically use it to separate proteins based on size. It's done in these gels, these vertical gels, and you basically send the protein traveling down vertically. What happens then in an electroblot is we kind of take things out of the gel and we transfer them to a membrane. So with SDS page, remember, SDS stands for sodium dodecyl sulfate. That's our detergent. It's going to help us unfold the protein and coat it with a negative charge. This is going to allow it to travel through the gel. And when it travels through the gel, it's going to get separated by size. The longer molecules, the bigger proteins are going to get tangled up more. They're going to travel more slowly. And so when you turn off that power, they won't have moved as far. If you were to go and you were to do like a general protein stain, a chromacy stain, you would then see bands for each protein in the gel, but you wouldn't be able to tell which band corresponded to what protein, just their relative sizes. If, however, you use a Western blot, well, now you can actually go and you could use antibodies to look for specific proteins. So we send the proteins traveling through the gel through SDS page, and then we're going to transfer them to a membrane to see how much of a protein is there. We don't want to try to probe directly into the gel for a few reasons. One is that these gels are really, really thin and they rip really easily. Two is that we're gonna be putting these membranes through a lot of washes and stuff. Two, we don't want things to like diffuse out of the gel. Three, I don't even know how well the antibodies would be able to find the proteins in the gel. Lots of different reasons. We transfer it to a membrane instead. So we go down in our SDS page and out in our electroblot. And so sometimes there's different formats of electroblot. This is like a wet transfer. There's also like a dry transfer, semi-dry transfer. There's basically a lot quicker ways. Like in my postdoc lab, we had this like really cool device that did the Western blot in like 10 minutes, and it was awesome. But this is the more classical way, and it's long and tedious and uses like methanol or ethanol and stuff. So there's a lot of waste you have to deal with, and it's not very fun. But don't worry, we're not going to be doing that in the lab. So the basic overview of the Western blot, we start with that SDS page and then we transfer it. Once we've transferred, well, we're basically transferring it to this membrane that is basically kind of like duct tape for proteins. It'll just bind to any protein. So it's going to bind to all those proteins as they come off the gel. And then we want to probe for it with an antibody. Well, do you recognize what might be a potential problem here? We've just been talking about how antibodies are just proteins, right? So we don't want our antibody to stick to the membrane just everywhere, because then we get signal everywhere. If we go and look where the antibody's bound, we're just going to see that the antibody's bound everywhere, and that would not be helpful. So instead, we block the membrane. Basically, we coat it with a generic protein. So often this is going to be actually like you use like skim milk. There's a bunch of the protein casein. Basically, you just need a generic blot protein to coat the sites that aren't bound by your protein, so that the antibodies can't bind to them. After you do that, then you send in the antibody that recognizes your thing. We call this the primary antibody, which is going to bind the specific protein you're looking for. Then after a bunch of washes, you send in a secondary antibody. Now, the secondary antibody is going to recognize the primary antibody. More on this in a second. And it's going to have something that's detectable. So this could be like a fluorophore, some sort of enzyme that can make colored or fluorescent chemiluminescent product. Some way that this is going to allow us to detect a specific protein. So going back to the idea of primary and secondary antibodies, basically, remember how I said that antibodies had that generic part and that unique part? Well, the generic part is only generic for the animal that made the antibody. And then basically what can happen is you can use a secondary antibody that recognizes that generic part from the first antibody. And then the secondary antibody is conjugated. So remember, conjugated just means it's like attached to something detectable. So for example, your primary antibody might be like rabbit anti-something, and then your secondary could be goat anti-rabbit. Why do we do this? Well, there's various benefits. One is that it's going to allow you to have signal amplification. So basically, if you only have a single antibody bound to your antigen, to your protein, Especially if you're using a monoclonal antibody, there's only one epitope per protein. So only one antibody can bind to one protein. So that might not have a strong enough signal if you were just using a primary antibody. But by using a secondary antibody, you can then have multiple secondary antibodies binding to that primary antibodies, allowing for signal amplification. But probably more importantly, cost. So basically, these antibodies, even if we're using an epitope tag, these antibodies are still expensive. And so if it would be even more expensive if you then had to have each of those antibodies conjugated to something detectable. So instead, what happens is that because a bunch of people all use mouse antibodies, or a bunch of people all use rabbit antibodies or goat antibodies or different antibodies, then what can do is basically you can get secondary antibodies that are conjugated to something detectable. And because lots of people will want those, the companies can sell them like a lot cheaper because they can sell more of it. So they make them cheaper. And so you can get these secondary antibodies. And by having them be cheaper and more people want them, you can actually get secondary antibodies with a bunch of different detection methods. So these could be fluorophores. So remember, a fluorophore is just something that absorbs light of one wavelength and lets off light of a different wavelength. They can also be enzymes like coarse radish peroxidase, HRP, which can then transform a invisible chemical to a colored one, or one that is going to give off light. So various methods using secondary antibodies conjugated to various things that you can detect. One way or the other, you're going to be able to then go and look and see where that antibody was bound, showing you where the protein was. So remember, like our chromatography staining, that's going to show us all proteins, whereas the Western probing is going to show specific proteins. So you can see in this example, yes, I have the protein here, but then I have lots of other proteins. And only when I actually go and do the Western blot can I see which of those proteins were the ones I was specifically looking for. Because especially if the proteins are similar size, they're going to overlap. And so a single band might correspond to actually multiple proteins. So we're going to look at an example of how to interpret a Western blot. But first, one other thing is that typically people also do a Western blot for some sort of housekeeping control. So basically, to allow for normalization. So if you were to look at two different nearby lanes, say these were two samples from two different cells, could we say that this cell expressed more of the protein than this cell? Not unless we have some sort of way to know that we actually had the same amount of both of these cells. We could have just loaded a lot more of this sample than this sample. And so we need some sort of internal control to normalize to. Often these are going to be like GAPDH, actin, or tubulin, various proteins that cells typically make in similar amounts, and they make a lot of it. And this is going to then allow us to basically normalize to the levels of those in order to directly compare different lanes. And so people do something like this, where they're looking for these various things. They're looking for this protein AKT. People often probe for multiple things, so not just the housekeeping control and what they're one thing, but also another thing. So here they're looking for this protein called AKT, and here they're looking for a specific phosphorylation site of AKT. Because remember, the antibody is just recognizing that small little epitope. And so if that epitope is if you can recognize a phosphorylated epitope, and so that's what this antibody was made to recognize. And so you can see that, well, although they have similar levels of AKT, some of them have more of the phosphorylated version than others. And by comparing them directly to the levels of GAPDH, which we can see are very consistent between them, then you're actually able to somewhat quantify the levels. And so you will see often people will do these Western blots and then normalize to their control to show you, or normalize to the GAPDH in order, or whatever housekeeping gene, in order to then go and show you how we can compare the actual amounts. In an ideal case, you'll see a single band, but When you see things like this, where basically they're just showing you slices of the gel, you really have no idea what's going on in the rest of the picture. And it could be something like this. With antibodies, we often get a lot of non-specific bands. Sometimes we have to really weak or no signal, high background, all sorts of different problems. But one of the main problems is this non-specific band formation. And so often you have to have a bunch of controls in order to show that your antibody really is recognizing the thing that you want. And then you have to go and you have to like actually, although you can get away with showing just these like strips in the paper, you're often then in the supplemental or extended data, they'll make you show like your actual raw gels with all of their different beautiful Western blots. So that's one way we can use antibodies. Another way is co-immunoprecipitation or co-IP. So immuno, we're talking about using our antibodies here, and precipitation, basically like pulling out. So in a co-IP, which is often also called like a pull down, a thing of interest, so a specific protein or something is used as bait, and then we want to see what it's bound to. So we call that our prey. So what we can do is basically we take a sample. So this could be a cell lysate. So you broke up in a cell. You want to see what a protein or what a specific protein is interacting with in a cell. And then what you do is you use antibody conjugated beads to capture that thing and the stuff that it's bound to. You wash off the non-bound stuff and then see what it's bound to. So similar in concept to our affinity chromatography, except here we're just kind of like using a small little sample. Often these are like magnetic beads, and we do it in this sort of like you have these little magnetic beads in these tubes and you put it on a magnetic rack and it pulls the beads and then you can just like pipette out the liquid. It's really, really cool. It makes me really happy to use, except when the beads like when the stand's not cooperating and stuff. But anyway, having this magnetic form is really, really handy. You're then able to isolate your thing, wash off other things, then elute your protein and analyze it. So in order to elute it, there are various ways. Some methods you just kind of like heat it up and let things fall off, because remember, antibodies are proteins. In some cases, you compete it off with a competitor, various strategies. One way or the other, you're then able to go and look and see, okay, well, what was my bait bound to? So often in order to look, they're actually going to do a Western blot. So you'll see Western blots commonly associated with a co-IP. You can also use mass spectrometry, mass spec. Basically there, they just chop up all the proteins into little pieces, send those proteins traveling through this machine that's going to kind of measure the mass charge ratio of those pieces. Then they compare those values that they get to a database in order to figure out what proteins were in the mixture. So that's kind of like an unbiased approach that's going to show you everything that was bound to it. Whereas if you do a Western blot, remember with the Western, you kind of have to know going in what you want to look for. So if you don't know that it might be bound, you're not going to be able to tell if it was. Just a quick note, so co-IP is often called a pull down, but there's also other versions of a pull down that that don't use antibodies. So you could, for example, use a protein with a strep tag, then use like streptactin resin, streptactin beads instead of using an antibody. And in this case, or you could use like glutathione to bind to a GST tag protein. So there are various other forms of pull down that might not specifically use antibodies, but they use the same basic concept of being able to isolate one thing based on some sort of feature and then see what it's bound to. So let's talk a little about how we interpret a pull down blot, and then we'll look at some actual examples. So typically what you'll see is on the left, you'll see an input lane. Basically, this is just showing you what they put in. So this is important because, well, if there wasn't any of your bait in the sample, how are you going to see if it's in the pull down? Or if there was a lot of one protein in the input, and then you see a little bit of it pulled down, then that's a lot less significant than if there's a little tiny bit of it in your input, and then like all of that is still in your pull down. So the input's important to kind of be able to compare, okay, well, how much was there to start with versus how much of it actually gets pulled down. It's also important to have a negative control. So this might be like a pull down with naked beads. So with beads without antibody bound, or with beads with an antibody for something that you know is not in the mixture, such as something like for our mouse, or targeting a different protein that you're not looking for, various things like this. So this is important to show that, yeah, your antibody is actually specific and your washes are sufficient. So we'll talk more about washes in a second, but that's what you'll see. So you have your input, what's going in, your negative control, and then your pull down. Sometimes this is like labeled like IP. And basically this is going to be the stuff that was pulled down when you isolated that bait, the prey. So you'll see the bait as well as whatever prey it was bound to. Okay, so let's get some practice interpreting one of these blots. What you'll typically see is something like this. On top, you'll see this sort of table with pluses and minuses. And then on the left, there's going to be, you'll see the input. And on the right, you'll see the IP. So this is your pull down. And so here they're specifically telling us that they're doing a pull down against HA. Now over here, they're saying that in the samples, basically the samples differ. So this one, flag glue A1, this is a receptor, basically that has an epitope tag, a flag tag. And then they also have HAPRRT1, which is some sort of other protein that has this HA tag. So they have two tagged proteins. They're going to be doing their immunoprecipitation against the HA. So basically they're going to be pulling out this HAPRRT1, and then they're going to be doing an antibody again. They're using an immunoblot, so a Western blot, both against the flag tag, which will tell them if glue A1 is there, as well as against the HA, which will tell them if PRRT1 is there. And so in one of their samples, they have the flag glue A1, but not the HAPRRT1. And in the other sample, they have both of them. And so you'll see the same, you should see the same like thing in the input and in the IP in terms of the table above, what they're putting in. But then what they see with their immunoprecipitation might be different. So whenever you're doing an immunoprecipitation against something, you should see that something in your immunoprecipitation if it was present in your input. So basically here, what we're saying is, okay, we're going to be doing our IP against HA. In this first sample, we don't have any HA, so we shouldn't expect to see any of it in our IP. And in fact, we don't. So that's good. Kind of like a negative control. Okay, now what's going to happen is if we think about glue A1. So we can see that in both of these samples, we have our glue A1, but are we going to see it in our pull down? Well, we're pulling down the HA. So the only way that we'll see glue A1 is if it's actually interacting with our HA. And so do we see glue A1? Well, we have to look and see, okay, they're immunoblotting against the flag, because that flag is going to tell us about glue A1. So we can see here that, yes, we see that. We see the HA, which is good because, well, that's what we're pulling out. And so we want to make sure we can see it, that we actually are pulling it out. And we also see this glue A1. And so this is telling us that these two proteins were interacting. What about in this case? Here we're looking at a different receptor, this glue A2, but the rest is the same. How do we interpret this? Are they interacting or not? Right, this is similar to before. We can see that they're interacting because we see both of them in our pull down. Let's look at a couple more examples. So here they're looking at glue A3 and glue A4. Yeah, they look pretty similar to before. We can see that they're both interacting. We can also see that they have some non-specific bands in here. And so they need to be a little careful with their interpretation, but they still have a lot more signal than they have with this non-specific signal. So yes, we can say that they do seem to be interacting. Okay, what about this one though? This glue A1 with this PRRT2. So now we're looking at a different protein interacting. Do we see interaction here? Yeah, so basically there's a little tiny bit, but this interaction must not be nearly as strong. There might be a weak association, a weak interaction, but not nearly as much as in our other cases. So finally, they have this other negative control where here they want to see, okay, well, is PRRT1 just like randomly binding everything? And so they choose this other random protein, NRXN1, which it shouldn't be binding to. And they want to see, okay, well, if the binding is really, really non-specific, then we would be expecting to see NRXN1. Or if our wash conditions were so bad that basically things were just all sticking together and everything was there, we would expect to see it. But if things are good, if our binding is specific, then we don't expect to see NRXN1 when we pull down PRRT1. So in fact, when they pull down PRRT1, well, now what they see is basically they're not going to see NRXN1. So they pull down the PRRT1 to the HA tag, and then they do a Western blot to the flag tag to look for NRXN1, and they don't see it. So this is how you would interpret something like this. How do you think their results might have been different if they had done a higher salt concentration? How might those salt concentrations of the washes affect what gets detected? Think about what you know about intermolecular forces. Right, the interactions are going to be weaker. And so we might see fewer detections, we might see fewer proteins detected, and we might see lower amounts of those that are detected. Remember that even for non-permanent charges, even non-ionic interactions, we can still think about them as on some level being partially charged based. Well, what's going to happen is if you have high salt concentrations, you're going to have lots of your ions floating around. And those ions are kind of going to create this fogginess, this electric fogginess that's going to kind of distract the binding partners, compete for those binding interactions. It's going to make it harder for those charges to feel one another. It's going to make those interactions weaker. And so the higher the salt wash is, the stronger the affinity has to be for things to stay bound. Those are the main methods I wanted to talk about. Another one is ELISA. So our enzyme-linked immunosorbent assay. So enzyme-linked because often we're going to be using one of those secondary antibodies that's attached to like HRP or some other enzyme, where we can use some sort of like chemiluminescent or colorimetric readout. So something that changes color, something like this, or something that maybe gives off fluorescence, chemiluminescence, things like this. Sometimes it might be directly conjugated to a fluorophore instead of an enzyme, but we typically still call it an ELISA. Often it's used to detect either like antibodies or antigen in a patient's blood or in some other sample. There are various ways that ELISA works, but in all these cases, you kind of have a plate. And on this plate, you have like an antibody attached, or you could actually have an antigen attached to the plate. Then what's going to happen is you flow something else over. That something else, if it contains something that binds to that antibody that's attached to the surface or binds to the antigen that's attached to the surface, then it'll stick. And then you can kind of use those various detection methods in order to see if the thing was stuck. So it differs from a Western blot in that this is taking place in like the wells of a dish, rather than like on a membrane, and you don't have to separate things first. But one of the downsides is that you're not seeing anything about like the size of the things. And if we think back to that Western blot with it was like all those non-specific bands, well, now if you see a signal in your ELISA, is it really coming from the protein you think it is? It might not be. So you need to be really, really careful and have good controls so that you can subtract out the background. There are also benefits of Western blots over ELISA in things like seeing the different sizes can tell you if there are different forms of your protein, and things like that. So lots of cool things about Western blots and co-IPs and stuff that you couldn't get from an ELISA. But an ELISA does have benefits, such as being able to test patient samples and things like this. And so here's an example of an ELISA that was used to quantify the human antibodies in mice blood sera. And so basically you can see that they kind of set up their plate. Typically people do like different titers or different like dilutions of the blood sample or of the serum sample, whatever sample, to try to get like a quantification. And so then you'll see something like this, where they can then measure the amounts of the antibodies. Epitope tags, you're going to see these a lot. HA flag, MIC, V5, HIS, all those good things, these short little tags that we can stick onto the end of a protein to allow us to use antibodies more easily against them. And so why do we want to use antibodies? Lots of different reasons, including Western blots, where we're looking for specific proteins after we run an SDS page gel. Co-IPs, where we're looking to see what proteins are bound to what proteins a protein is bound to, or things like an ELISA, where we're looking to see something similar to a Western blot, but in the wells of a dish. So hope that gave you an overview, and then in class, we'll look at a bunch of examples of how we actually interpret the data. And this will help you when you're trying to interpret the data in your papers, as well as in all the other papers that you'll encounter in what I hope will be a life as a scientist, or at least just a person that can appreciate the value of biochemistry and be able to continuously learn throughout your life and take advantage of the biochemical literature without just being like intimidated by it.